Hello viewers, I am Dr. Robiul. I work as a lecturer of pathology in a medical college hospital and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is antibodies. This is the third lecture of our ongoing immunology lecture series. Since this is a big topic, so I will upload two videos on antibodies. In the first video, we will learn the definition of antibody, the structure of immunoglobulins. We will also talk about the classes of antibody and their important functions, polyclonal and monoclonal antibodies, concept of isotypes, allotypes, and idiotypes. Then in the second video on antibodies, we will learn about immunoglobulin genes, immunoglobulin class switching, hybridoma cells, and how monoclonal antibodies are produced from hybridoma cells. Okay, a lot of topics, so let's begin. We will start our discussion by talking about the definition of antibody. So how can we define antibodies? As we can see, antibodies are globulin proteins or immunoglobulins that react specifically with the antigen that stimulated their production. We can also define an antibody as a protein substance that is produced as a result of antigenic stimulation. Now always remember, antibodies make up 20% of plasma proteins. Blood contains three types of globulins according to their electrophoretic migration rate. They are alpha, beta, and gamma, and always remember antibodies are gamma globulins. So now that we have defined antibody and also talked about some introductory points, now let's move on and talk about the structure of immunoglobulins. So as we can see, antibodies or immunoglobulins are glycoproteins and they contain light and heavy polypeptide chains. Light chains have a molecular weight of about 25,000. Heavy chains have a molecular weight of 50,000 to 70,000. The simplest antibody molecule will contain 4 polypeptide chains and it will be Y-shaped, and again the 4 polypeptide chains will include 2 heavy chains and 2 light chains. The molecular formula will be H2L2. Note that here H is denoting heavy chain and L is denoting light chain. Always remember, an individual antibody always contains identical heavy chains and identical light chains. The light chains and the heavy chains are subdivided into variable and constant regions. Again, the regions are made of three-dimensionally folded, repeating segments that are called domains. Each domain is approximately 110 amino acids long. A light chain contains 1 variable domain and 1 constant domain. Most heavy chains contain 1 variable and 3 constant domains. For example, immunoglobulin G, D, and A, they have 1 variable and 3 constant domains in their heavy chains. However, immunoglobulin M and immunoglobulin E have 1 variable and 4 constant domains in their heavy chains. Since they have 1 extra constant domain, so it makes them longer and heavier compared to immunoglobulin G, D, and A. Now this image is showing the structure of a simple immunoglobulin molecule. On the top we can see the amino terminal end of this immunoglobulin, and on the bottom we can see the carboxy terminal end. We can also see that this immunoglobulin is made up of 4 polypeptide chains. These are the light chains, they are smaller, and the remaining two are heavy chains, they are larger. We can also see that these four polypeptide chains are bound together with the help of interchain disulfide bonds that are drawn here in green color. Note that part of light chain and part of heavy chain are drawn in purple color, and these are the variable regions of immunoglobulin. Now, you may be asking me, Dr. Robil, why are they called variable regions? They are called variable regions for their variation in amino acid sequences between immunoglobulins synthesized by different B lymphocytes. And we can see that this is the variable domain of light chain, this is the variable domain of heavy chain. Similarly, in orange we can see the constant domain of light chain as well as the constant domains of heavy chain. Now let's talk about the function of variable region. In both heavy and light chain, the function of variable region is antigen binding. Antigen-antibody binding involves electrostatic and van der Waals forces and hydrogen and hydrophobic bonds. What is the function of constant region? It has several functions. They will include complement, activation, and binding to cell surface receptor. But we are talking about the constant region of heavy chain. The function of constant region of light chain is in fact unknown. Now, in your textbook, you will also find a term called hypervariable region. So what do we mean by that? Always remember, both heavy and light chains have three extremely variable amino acid sequences at the amino terminal end of their variable regions that form antigen binding sites. These three extremely variable amino acid sequences are known as the hypervariable region. 5 to 10 amino acids in each hypervariable region form the antigen binding site. So here we can see hypervariable regions of heavy chain that are shown here in blue lines in the variable region, and these are the hypervariable regions of the light chain. So let's talk about light chains and heavy chains now. Light chains are of two types according to amino acid differences in their constant region. They are kappa and lambda. Kappa is encoded on chromosome 2 and lambda is encoded on chromosome 22. Both types can be found in all classes of immunoglobulins. The last bullet point is also very important. Always remember that any one immunoglobulin will contain only one type of light chain. Regarding heavy chains, always remember that they are distinct for each of the 5 classes of immunoglobulins. For example, in case of immunoglobulin G, the heavy chain is gamma. In case of immunoglobulin A, it is alpha. For immunoglobulin M, it is mu. For immunoglobulin E, it is epsilon. And for immunoglobulin D, the heavy chain is delta. All 5 types are encoded on chromosome 14. So now let's talk about some very important topics for our examination, and that is the FAB and FC fragments. So what do we mean by these two terms? If an antibody molecule is treated with a proteolytic enzyme like papain, the peptide bonds in the hinge region will get broken. This will produce 2 identical FAB fragments and 1 FC fragment. FAB fragments carry the antigen binding sites, FC fragment is involved in complement fixation, attachment to various cells, and placental transfer. So in this image, this is the hinge region of the immunoglobulin, and it is rich in proline and it provides flexibility to the immunoglobulin. Now, if this immunoglobulin was treated with proteolytic enzyme papain, that will break down bond in this hinge region. As a result, the immunoglobulin molecule will be broken down into 3 fragments. 2 will be identical FAB fragments. So this will be one FAB fragment, and this will be another FAB fragment, and the third fragment will become FC fragment. So now that we have talked about the structure of immunoglobulin, now let's move on and talk about the classes. There are 5 classes of antibodies or immunoglobulins. It is based on differences in their heavy chain, and we can remember them by using this mnemonic GAINED. So the 5 classes of antibodies are immunoglobulin G, A, M, E, and D, and we can remember them by using this mnemonic GAINED. So let's talk about them one by one, and we will start by talking about immunoglobulin G. It is composed of 2 heavy chains and 2 light chains. The chains are linked by disulfide bonds. Immunoglobulin is divalent and contains 2 identical antigen binding sites. It has 4 subclasses. They are immunoglobulin G1, 2, 3, and 4. Most common is immunoglobulin G1, that is 65%. Always remember that immunoglobulin G1 and G3 are more effective opsonizers compared to immunoglobulin G2 and G4. So here we can see an image showing the structure of immunoglobulin G, and we have already discussed the amino terminal end, carboxy terminal end, and interchain disulfide bonds. Now always remember, immunoglobulin G is the only antibody that can cross the placenta. Its FC portion can bind to FcRn receptors on the surface of placental cells. FcRn receptor can transport maternal immunoglobulin G across the placenta into fetal circulation. That's why immunoglobulin G is the most abundant immunoglobulin in newborns. Always remember, immunoglobulin G can activate complement. Immunoglobulin G and immunoglobulin M, these 2 are the only antibodies that have the capability to activate complement. Immunoglobulin G can opsonize or enhance phagocytosis. The surface of phagocytes contains receptors for the gamma H chain or the gamma heavy chain of immunoglobulin G. That's why immunoglobulin G can enhance phagocytosis. So now let's talk about immunoglobulin A. It is the main immunoglobulin found in secretions like saliva, tears, respiratory tract secretion, intestinal tract secretion, genital tract secretion, and IgA is also found in colostrum. Now, secretory immunoglobulin A are dimers, and they are composed of 2 H2L2 units plus 1 unit of J chain and secretory components. On the other hand, serum immunoglobulin A are monomers, and they are composed of single H2L2 unit. So here we can see secretory immunoglobulin A. Note that it is a dimer, and the J chain is shown in red color and the secretory component is shown in blue color. So what do we mean by secretory component? It is a polypeptide made by epithelial cells. It helps in passage of immunoglobulin A to the mucosal surface, and it also prevents its degradation in the intestinal tract. Moving on to immunoglobulin M, it is the main immunoglobulin for primary immune response. It is found as monomer on surface of all B lymphocytes, where it acts as antigen binding receptor. It is also found as pentamer in the serum, and there it is composed of 5 H2L2 units plus 1 J chain. Now, immunoglobulin M pentamer has obviously 10 antigen binding sites, and we have already mentioned that immunoglobulin M can fix complement. Moving on to immunoglobulin E, it can mediate immediate hypersensitivity reaction. It is the main defense against helminth or worm infection, for example, Ascaris, Trichinella strongyloides, and also against hookworm infections like Nicketer's and Ankylostoma. Worms are too large for phagocytes to ingest, so they are killed by worm-destroying enzymes that are released by eosinophils. And immunoglobulin E has important roles here. Immunoglobulin E, specific for worm protein, will bind with receptors on the surface of eosinophils, and that will trigger cellular cytotoxicity to kill the worms. Moving on to immunoglobulin D. Now, it has no known antibody function, but it is present on surface of many B lymphocytes, and it may act as antigen receptor. So here we are summarizing the important functions of immunoglobulins or antibodies. They neutralize toxins and viruses, they opsonize microbes, they can activate complement, they can prevent attachment of microbes to mucosal surface, and they also have catalytic functions. Now, this table is very important for your examination, and in this table we are summarizing the important functions of the different classes of antibodies. So, what are the functions of immunoglobulin G? You have to remember that immunoglobulin G opsonizes bacteria and fixes complement. We also have to remember that immunoglobulin G is the only antibody that can cross the placenta, and that's why it is in newborns. Immunoglobulin G can also neutralize bacterial toxins and viruses. What are the functions of immunoglobulin A? Secretory immunoglobulin A can prevent attachment of bacteria and viruses to mucous membrane. Immunoglobulin M can fix complement, and immunoglobulin M can also act as antigen receptor on the surface of B lymphocytes. What are the functions of immunoglobulin E? We have already mentioned that it mediates immediate hypersensitivity, and it also provides protection against worm infection. And the function of immunoglobulin D is uncertain. So now that we have talked about the various functions of antibodies, now let's move on and talk about polyclonal and monoclonal antibodies. So what do we mean by polyclonal antibodies? These are formed by several different clones of plasma cells in response to typical antigens. Since they are made by different clones of plasma cells, they are heterogeneous. And what do we mean by monoclonal antibodies? They are formed by single clone of plasma cell, and that's why they are homogeneous. Example will include antibodies that are produced in certain plasma cell tumor, for example in myeloma. So now that we have talked about the concept of polyclonal and monoclonal antibodies, now let's move on and talk about the concept of isotypes, allotypes, and idiotypes. So what do we mean by isotypes? These are antigenic differences in their constant regions. All isotypes are found in all normal humans. For example, the 5 classes of immunoglobulins, they are isotypes. What do we mean by allotypes? These are additional antigenic features of immunoglobulins that differ among individuals. And what do we mean by idiotypes? These are antigenic determinant formed by specific amino acids in the hypervariable region. So this concludes our first video on antibodies. I hope this video was helpful. If you like my videos, do comment, share, subscribe, and let me know. And for my students, I will also recommend you to go through your textbooks to know more information. I will upload the second part of this topic hopefully within a week. Until next time, take care and stay blessed. Thank you.